The goal of this procedure is to produce recombinant adeno-associated viruses, or RAAVs, for effective in vitro or in vivo transgene delivery. This is accomplished by first transfecting HEC293 cells with DNA plasmids encoding the transgene and viral genes. The transfected cells are then scraped and lysed to harvest the assembled RAAV particles. Next, the RAAVs are purified from the cell lysate through heparin column purification. The final step is to titer the viral stocks. Ultimately, the result shows strong and persistent virus-mediated transgene expression through immunofluorescence microscopy. At this point, the RAAVs are ready for in vitro or in vivo applications.
The main advantage of using our technique over other purification methods, such as the cesium chloride gradient purification, is that we do not require to use an ultracentrifuge or toxic materials. We also yield higher and purer viral vectors.
Prior to the start of this protocol, prepare high-quality plasmid DNA stocks and screen for integrity, as described in the written protocol. To begin, plate 2 80% complement 150 cm² flasks of HEC293 cells into 5 15 cm diameter NUNC tissue culture dishes. Culture cells in standard DMEM with low glucose containing 10% fetal calf serum. Approximately 48 hours after plating, on the day of transfection, cells should be 70 to 80% complement. 3 hours before transfection, remove the DMEM and replace with Ice Cove's modified Dolbachow's medium or IMDM, containing 5% fetal calf serum. Prepare the transfection mixture for a single batch of virus, as outlined in the written protocol. In a Class 2 tissue culture hood, sterile filter the transfection mixture into a 50 ml tube. Whilst vortexing the solution, quickly add 13 ml of 2 times Heapy's buffered saline. Replace the lid of the 50 ml tube and continue to vortex for 15 seconds. Leave to stand for 1 minute and 45 seconds. A white precipitate should form. Gently add 5 ml of the transfection solution to each 15 cm tissue culture dish. Swirl the plates to mix. After incubating for 16 hours, remove IMDM, replace with DMEM, and continue to incubate the cells. To harvest the RAAVs, remove media from cell culture plates 72 hours after transfection and discard. All waste should be treated with Virkon solution or other suitable disinfectant. Gently wash the cells in warm 1 times phosphate buffered saline. Add 25 ml of warm PBS to each plate and gently remove the cells with a cell scraper. After collecting the suspension in 50 ml tubes, pellet the cells at 800 times G for 10 minutes. Discard the supernatant and resuspend the pellet in 10 ml of 150 millimolar sodium chloride buffer per tissue culture plate. Split the suspension into 2 50 ml tubes. Next, lyse cells with a freshly prepared solution of 10% sodium deoxycholate. Then, add benzonase nuclease for removal of nucleic acids. After mixing the tubes thoroughly, incubate at 37 degrees Celsius for 1 hour. Following incubation, remove cellular debris by centrifugation at 3000 times G for 15 minutes. Transfer the supernatant to a fresh 50 ml tube, ensuring that all cell debris has been removed to prevent blocking of heparin columns. For purification of RAAVs, add 10 ml of sodium chloride buffer to a syringe and set up the high-trap heparin columns using a peristaltic pump, so that solutions flow through the column at 1 ml per minute. It is important to ensure that no air bubbles are introduced into the heparin column. Equilibrate the column with 10 ml of 150 millimolar sodium chloride buffer. Apply 50 ml of virus solution to the column and allow the solution to flow through. Then, wash the column with 20 ml of 100 millimolar sodium chloride buffer. Using a 5 ml syringe, continue to wash the column with 1 ml of 20 millimolar sodium chloride buffer, followed by 1 ml of 300 millimolar buffer, discarding the flow-through. Employ 5 ml syringes and gentle pressure to elute the virus from the column by applying increasing high salt tris elution buffers. Collect the eluate in a 15 ml centrifuge tube. Concentrate the vector using Amicon Ultra 4 centrifugal filter units with a 100,000 molecular weight cutoff. Load 4 ml of the column eluate into the concentrator and centrifuge at 2000 times G for 2 minutes. After repeating the centrifugation, the concentrated volume should be approximately 250 microliters. Discard the flow-through and reload the concentrator with the remaining virus solution. Add 250 microliters of PBS to the virus for a final volume of 500 microliters, and remove from the concentrator. As a final step, filter the vector through a 13 mm diameter 0.2 micron syringe filter. To titer the stock virus, prepare 18 polyl-lysine coated glass cover slips by seeding HEC293 cells so that they are 40 to 50% confluent. For titration of Cre-dependent RAAVs, use HEC293 cells that stably express Cre recombinase. Infect each well with serial dilutions of the RAAV vector as described in the written protocol, using 3 wells per dilution. After 72 hours, fix HEC293 cells by adding an equal volume of 4% paraformaldehyde to the medium for 10 minutes at room temperature. Following incubation, rinse the cells 3 times in PBS and once with distilled water. Then, using mounting media, place the cover slips onto the slides, cell side down. Count the number of transduced cells from the 3 wells that have the highest dilution factor but still contain infected cells. Find the average of these and multiply by the dilution factor to give the number of infectious units per microliter. HEC293 cells transduced with a virus-encoding enhanced green fluorescent protein are shown. Consistent titers of around 6 times 10 to the 6 infectious particles per microliter are generally achieved. Shown here are examples of stereotaxic injections of Cre-dependent RAAVs encoding green fluorescent protein into different brain regions of adult parvalbumin Cre transgenic mice. Viral green fluorescent protein expression is restricted to Cre-expressing neurons in different target regions. In this example, green fluorescent protein immunoreactivity is shown in the reticular thalamus and globus pallidus, the dentate gyrus, and the CA1 region of the hippocampus.
Once mastered, this technique can be performed in 7 to 8 days for virus production and 3 days for titering if performed properly.